Good afternoon and welcome to the USDA Human Nutrition Research Center on Aging's Fall Monday Seminar Series. Before introducing today's speaker, let me start with a little housekeeping. I'm sure that there'll be many questions today for our speaker. And to ask your questions, please use the Q&A icon located on the webinar toolbar. You can add questions at any time during the presentation as they arise. No need to wait until the end. And please to ask your questions, please use the Q&A icon and not the chat box. And now I have the pleasure of introducing today's speaker, Dr. Ana Maria Cuervo. Dr. Cuervo is the Robert and Renee Belfer Chair for the Study of Neurodegenerative Diseases and is Distinguished Professor in the Department of Developmental and Molecular Biology and the Department of Medicine at the Albert Einstein College of Medicine. She is also co-director of the Einstein Institute for Aging Studies. Dr. Cuervo's research is focused on understanding how altered proteins can be eliminated from cells and their components recycled. Her research has linked alterations in lysosomal protein degradation with different neurodegenerative diseases and shown that restoration of normal lysosomal function prevents accumulation of damaged proteins with age. Her lab has also pioneered studies demonstrating a tight link between autophagy and cellular mechanism. Dr. Cuervo has received numerous awards for her pioneering work, but to allow time for her presentation today, I'll only note two of her more recent honors: her election to the American Academy of Arts and Sciences in 2018 and to the National Academy of Science in 2019. And today, Dr. Cuervo will discuss her research in a presentation titled Selective Autophagy as a Geotherapeutic Target. And with that, let me turn the microphone over to Dr. Cuervo.
Thank you very much, Paul, and thank you, Jose, and all the members of the committee for giving me this opportunity to tell you a little about the work that we are doing. And as Paul mentioned, we are very interested on autophagy. And just to put it a bit on the context of aging research that I know many of you are interested in, among the different hallmarks or drivers of aging, our work mostly focus on this loss of proteostasis that occurs with age. So as you know, proteostasis refer to the need that the cells have of controlling the intracellular environment to make sure that every single protein in your cells is on the right conformation, on the right place, at the right time. And in order to make sure that that takes place in such a very crowded environment, and is the intracellular environment, the cells count on what is known the proteostasis network, that is basically chaperones that will monitor how is the protein conformation, where is it the protein, and when it has to move it around. And then if it is not possible to maintain the right conformation or the right place for that protein, the chaperones will eliminate those proteins through the proteolytic systems. And of course, you have major experts on the ubiquitin proteasome system in the center, so I don't need to mention that. But also there is another mechanism for elimination of proteins that is in the lysosomes, and that is what is known as autophagy. So we are mostly interested in autophagy, and traditionally, as I say, autophagy has been placed as one of the components that maintain cellular proteostasis. However, with the time, it has become clear that autophagy does more than just quality control. It participates by the ability of recycling everything that is inside your cell. It participates in cellular energetics. It modulates the function of mitochondria, has very nice described roles in maintenance of senescence or stemness, cell-to-cell communication, and the number of functions keep growing, and such as the growth of the different functions of autophagy, that in the new iteration of this beautiful, colorful chart of the drivers of aging, now autophagy has its own part of the pie, and it's considered that disabled autophagy is one of the drivers of aging. So, of course, autophagy doesn't require introduction. All of you know that it refers to the degradation of any kind of intracellular components in lysosomes, but I still wanted to keep an introduction slide to highlight that there are very different ways in which the cells can perform autophagy, and all these different ways coexist in any mammalian cell at a given time. So, for example, most of you are very familiar with this process of macroautophagy, in which basically portions of the cytosol are sequestered by this double membrane, fuse forming this vesicle, and then degradation occurs when this vesicle or autophagosome fuse with the lysosomes and the materials are degraded. And depending on the material that is being degraded, this process have different flavors. So if it degrades preferentially lipids, you can talk about lipophagy. If it degrades aggregates, aggregophagy, mitophagy, glycophagy. But basically the machinery, the core machinery is basically the same in all these types of autophagy. However, it's also possible to internalize materials directly by invagination of the vacuole, that is the equivalent of the lysosome in yeast, or in the case of mammals, by invagination of the membrane of the late endosomes, giving rise to these forms of microautophagy, because instead of this big vesicle, you just have these invaginating vesicles that pinch off and degrade. But in addition of these vesicle-mediated forms of autophagy, it is also possible to directly translocate proteins directly across the lysosomal membrane for degradation. And this process is known as chaperone-mediated autophagy. This process was identified by the latest Fred Dice, my postdoctoral mentor, actually in the Department of Physiology in Tufts. And this is going to be the process that I'm going to be mostly talking today. But if you have any questions about microphagy or macroautophagy, I'm more than happy to also entertain those ones. So what is this chaperone-mediated autophagy that I will be referring as CMA for short? So it's really a very simple process of protein degradation. Basically, it can only degrade proteins, so not organelles, but the proteins degraded by this pathway bear a pentapeptide, a sequence of 5 amino acids that is biochemically related to this motif. This it's not the exact amino acid composition, but if you are interested in knowing if your favorite protein or your whole proteome, how many of these motifs are there, I encourage you to use this free tool that we put online, and that you can, as I say, include your whole proteome and identify these type of sequences. And these sequences are important because when they are exposed, are going to be identified by a chaperone, the constitutive member of the HSP70 family, HSE70. And then the other major component of this pathway, in addition of the chaperone, is this receptor protein at the lysosomal membrane, the lysosomal associate membrane protein, type 2A or LAMP2A, that is really a splice variant of a single gene. The gene is LAMP2. The luminal region is identical for all the splice variants, and it's only the transmembrane and cytosolic tail that make it unique to the form of LAMP2A, B, or C, and only A is required for CMA. The process is really simple. Imagine that you have to degrade that red protein, so the chaperone is going to recognize that protein, bring it to the surface of the lysosomes, binds to LAMP2A, and now LAMP2A multimerized to create a translocation complex. While here, the protein is going to be unfolded, and assisted by another chaperone located in the lumen, the protein is going to be translocated and degraded. The process of CMA is constitutively active in almost every mammalian cell, but it's also inducible by the type of stressors that you can see here, and it's an important component of the cellular response to stress. With the years, we've been very interested on the regulation of this process, and I'm just going to highlight a couple of the regulators, just because most of this work has been published, but to give you an idea of the complexity. We have learned that a lot of the regulation occurs on the surface of the lysosomal membrane. So, for example, you have a tandem of GTP binding proteins that modulate the assembly of this translocation complex, how stable the translocation complex is, and then it's the assembly, because it has to deassemble in order to undergo a new process of binding and translocation of substrates. And then you have another axis that is modulated by the TOR2 complex and a phosphatase that through process of phosphorylation and dephosphorylation, it modulates the speed in which this complex assemble and deassemble continuously. So this one modulates the stability and this one the dynamics. There are also other regulators that signal from different parts in the cell. So, for example, there are negative nuclear signaling coming from the retinoic acid receptor alpha or from BMAL. And there is also positive regulation coming, for example, from the NF-κ pathway through calcium modulation or through oxidative stress, NFR2, upregulate this pathway. And sometimes other organelles also signal to this pathway. For example, we identify that peptides coming from the mitochondria, such as, for example, humanin, when they are released from the mitochondria, they can go to the surface of the lysosomes and stabilize the binding of substrates to the receptor. And under conditions of oxidative stress, is a way for the mitochondria to tell the lysosomes, get ready to do CMA because I'm going to cause some protein damage around here. With the time, we have also developed tools trained to identify and monitor this process. So we develop a fluorescent reporter that contains one of these sequences, and that when it's delivered to lysosomes, it makes them highlight in green. So if you have low CMA activity, the reporter will be mostly giving you cytosolic fluorescence. But when you activate CMA, this reporter will mobilize to lysosomes, and then you will see these dots. And in collaboration with Jose Javier Bravo Cordero, Mount Sinai, and Esperanza Arias at Einstein, Sushan in my lab developed a transgenic mouse model that expressed this receptor. So now we are able to monitor and visualize CMS. So for example, this is just a liver, and you can see the hepatocytes. We have staining lysosomes in red, and you can see wherever you see yellow, that means that this reporter has reached the lysosomes. And then we can do interventions. For example, this is a liver of a fed animal, and you can see here very few amount of PANTA. So you still have constitutive CMA, but it's not very high. But then if you remove nutrients, that is one of the stimuli for this pathway, you can see this very, very nice upregulation of CMA. So, of course, this is something that I describe even in my thesis, saying that CMA was activated through starvation, but we were doing all through biochemical studies and lysosomal isolation, and it's now very nice to see it in physiological conditions in an animal. So we have to take advantage of these animals to learn a little more about this pathway. And for example, Eve and Susmita in my lab identify that CMA is circadian. So for example, if they look in liver at different times, and remember these are mice, so this will be the light cycle is when they are less active, and the dark cycle is when they become more active. So you can see that at different parts of the cycle, you have different levels of activity. We found that the circadian occurs in almost every organ that we analyze, but if you look here, there is a shift or different times in which, depending on the organ, you get the maximal or the minimal activation of the pathway. So I'm showing you here liver in blue, heart in red, and kidney in green. And as you can see here, in all the organs, there is this circadian rhythm of CMA, but the times of maximal activation differ. We also found that the circadian regulation is under control of the clock machinery, because if we eliminate BMAL, that is one of the major components of the clock, as you can see here, you have CMA activity, but it doesn't cycle anymore, and it doesn't accommodate to the needs of the cell through the 12 hours light and dark cycle. And then to make it more interesting, we also found that in addition of the clock regulating CMA, CMA is able to degrade components of the clock timely. So, of course, you get the upregulation of the components transcriptionally, but then somebody has to bring them down, and part of this going down every 12 hours occurs through CMA. Also taking advantage that these animals were getting old, and we were during the pandemic and couldn't use them. So when we came back, we found that we have all this nice cohort of very old animals where we can now analyze how CMA activity changes with age. So we have been analyzing in young and in all animals, and we also include comparison of males and females. Because everybody in the lab has expertise in a different organ, everybody analyzed that organ, and something that we have learned is that CMA activity decreases with age in almost every organ that we have analyzed, but it's interesting that at the cellular level, there are differences on the dynamics and also across the organs, and also there are differences in the causes of this decrease with CMA depending on the organ and also depending on the sex. So just to illustrate a little, this is work led by Rabia, and this is work from Marian, and she was analyzing in young and old liver. So in this case, she's looking at hepatocytes, and when you look at the quantification, so in pink are the young animals, and in gray are the old animals. And as you can see, there are no gender differences in this case, so male and females have similar activity, but in both cases in hepatocytes, there is a decrease with age. However, if you, for example, look at the Kupffer cells, we identify that there are sex differences, so males have lower CMA activity than females, but in these cases, they are not affected by aging as the case of hepatocytes. And then we have gone through all the other organs and have found all these differences that the message, as I say, is that there is in general a decrease in CMA activity, but is very cell type specific and also sex modulated. We've been looking at what is the cause of this decrease in CMA, and one of the most general causes is a decrease in the levels of that receptor protein that I mentioned is limiting because it has to multimerize to create also the translocation complex. So for example, if you look in the liver of young and old animals, as you can see here, levels of LAMP2A decrease with age, but other lysosomal components do not decrease. So this seems to be very specific. We have similar decrease in LAMP2A in different parts of the brain at different ages. Even as you see, the kinetics are different. And just to show you another cell type, just so you can see that this seems to be very universal, these are hematopoietic stem cells. And as you can see here, as the animals get old, there is also a decrease in the levels of LAMP2A. So we have, as I say, we have become very interested in these changes of LAMP2A and CMA with age, and also in a series of age-related diseases. So to summarize a little what we have learned and work from our lab and many other labs have shown is that there are multiple challenges that when chronic and associated with aging are going to result in a decrease in CMA, and that seems to be related with some of these age-related disorders in the metabolism, at the level of protein quality control in central nervous system, or for example, also related to cancer, autoimmune disorders, or immunosenescence. But because of the topic of this series, I thought that I will focus today mostly on the changes with aging and in different metabolic conditions. So one of the things, just to show you that not only aging, but also dietary excess can contribute to decrease your CMA. In collaboration with Judy Slumer, we challenged animals with pro-atherosclerotic interventions. That is a combination of high-fat diets and PCSK9 delivered by ABV, as you know, that reproduce atherosclerotic plaques in animals. And as you can see here, using the same reporter, if we quantify the number of fluorescent panta in the aorta in normal conditions, compared to once you have challenged them with these interventions, you can see a dramatic decrease in CMA activity that we show that is once again due to decrease in LAMP2A. These are relatively young animals, and just that excess of lipids is enough to decrease CMA. But we also found that excess carbohydrates have a very similar effect. So for example, if you compare animals maintaining CHO diet with animals maintaining high carbohydrate diet, and this is just for 2 months, as you can see here, there is a reduction of the CMA activity in the pancreas, for example, and in liver, but we also found that the level of decrease varies from organ to organ, but it seems to be very generalized. As in the case of aging and in the high-fat diet, when we challenge with high carbohydrate diet, we also see that the major failure seems to be a decrease in the levels of this LAMP2A, in this case, both at the transcriptional level, as you can see here, for example, in the liver and in the adipose tissue, in the animals that have been fed this high carbohydrate diet, but also at, this reflects on the protein level, as you can see here. And once again, if you compare with another lysosomal component, the changes seems to be preferentially in this LAMP2A receptor that you need for CMA. So we are also very interested in trying to study CMA in humans, and unfortunately, we cannot put these reporters in people yet. So what we have been doing is to creating algorithms that allow us to infer what will be the final outcome on CMA by using the expression or the levels of proteins of the different components of the network. And this is work from Mathieu and Adrian in the lab, that basically by giving weight and directionality to each of the regulators of this pathway based on our previous 20 years of experimental data, they were able to generate an algorithm that produce a CMA score. It's a predictive index, but we found very interesting that if you compare this index in young, healthy individuals, and these are two different regions of the brain, you can see how in age, this is not disease, this is just healthy individuals, you can see that there is a gradual decline. Or for example, I was just talking about aorta. So if you look in the aorta, individuals at different ages, there is a gradual decrease in the expression of this transcriptional network of this CMA score. We are using that score also in the context of disease. So just to continue a little with this cardiovascular situation. So if you compare, for example, the CMA score in individuals with stable plaques or individuals with unstable plaques after a cardiovascular insult, you can see that there is lower CMA activity in the case of the unstable plaques. And actually, this is due also to a decrease in levels of LAMP2A, to the point that the levels of LAMP2A at the moment of the first cardiovascular incident seems to be predictive of whether there is going to be a second event. And then also in the case of the excess of carbohydrates, so in the case, for example, of diabetics, we have also been analyzing how is the CMA score, and this is work from Adrian in the lab, and he have shown that diabetic individuals have a lower CMA score, the same that has been shown in experimental mouse models of diabetes, and once again is related to a decrease in LAMP2A, in this case more pronounced at the level of protein. And what we start to find interesting, again, this is just predictive, but we found very interesting that if you try to correlate, for example, CMA score in people with their score or their risk for metabolic syndrome, you can see that the better is your metabolic, or the better is your CMA score, the lower seems to be your risk for this high metabolic syndrome score. So as I say, I know that many of you are interested in human studies. We don't do human studies, but I think this could become an interesting tool to at least predict if there is any correlation between CMA activity in people or the conditions that you are analyzing and the pathology. So based on our studies, and I hope I have convinced you that CMA activity decreases with age, not only in experimental mouse models, but also in normal healthy population. We are now very interested in addressing what are the consequences, because I can be very excited about this pathway, but maybe it's not so important after all. And if we convince ourselves that this pathway is important, whether we can do something about it and we can modulate it in order to slow down aging, and mostly the severity of age-associated diseases. So to study these consequences of reduced CMA, we decide to, based on the data that we have generated that suggests that a decrease in the levels of LAMP2A is almost a universal event that we found in all the contexts of aging. So we decide to go extreme and say, okay, let's get a young animal and basically eliminate only this LAMP2A. It was not as trivial as it sounds because it's an splice variant. So we messed up many times until we got it right, and we were only at eliminating LAMP2A, and we have normal levels of B and C. And then, of course, once you have a flux gene, this becomes a zoo. Now everybody in the lab is interested in eliminating CMA, specifically in their organ of interest. So we have liver, different parts of the brain, different cell types in the brain, skeletal muscle, adipose, hematopoietic, you name it. Every time that I go back to the lab, we seem to have a new animal. And then we also have an inducible variant where systemically we can eliminate CMA at different times. So, of course, I complain a lot because of having to pay for so many animals, but I think at the end it's worth it because we have realized that there are very organ-specific functions of CMA that we will have never identified if we will have not done this whole collection of animals. So what have we learned after looking at all these animals? So something that we have learned, okay, I'm not sure what is happening. Okay, yeah, so something that we have learned is that there are some functions of CMA that are common to all the components of the proteostasis network. So as many other components, the proteasome, macroautophagy, CMA contributes to protein quality control. So when you have oxidized proteins, acylated, damaged proteins, they can be selectively eliminated by CMA, the same way that you can eliminate them by the proteasome. And also that activation of CMA, because it breaks down proteins, can contribute free amino acids to replenish the amino acid pool. But as I said, these are functions that are common for all the proteostasis mechanisms, and actually one can compensate for other on these functions. However, something that we also learn is that CMA does not only degrade damaged proteins, it degrades fully functional proteins in order to terminate their function. And is this remodeling of the proteome what really confers particular unique functions to this pathway. So in a way, it's very similar to the proteasome. All of you are familiar how the proteasome, by degrading, for example, cyclins at different times, allow progression of the cell cycle. So in this case, it's the same. Our labs and others have shown that by timely eliminating, for example, glycolytic enzymes or lipogenic enzymes or lipid-binding proteins, it can modulate the flux through these different metabolic pathways. But it's not only metabolism. By degrading transcription factors, it can contribute to modulate transcription or translation in other conditions. The Mathian lab has identified how by degrading specifically blockers or T-cell activation, now it can promote T-cell activation. We have shown that can control also the cell cycle by modulating levels of CHEK1. And I just mentioned how it can control circadian by timely degrading these components of the clock machinery. So the idea is that whenever we have, or whenever think about the consequences of CMA, part of them might be related with damage accumulation and poor quality control. But as I mentioned, in many cases, this can be compensated by other proteostasis mechanisms. But the phenotype in most cases is related to this inability to timely eliminate these particular proteins to terminate their function. So just to give you some examples, for example, in the case of the liver, when we eliminate CMA in hepatocytes, we have seen that that has major consequences both in glucose and in lipid metabolism. So for example, if we eliminate CMA in liver, the first thing that we notice, these are wild-type animals, these are knockouts, is that if you stain for glycogen, these animals have very low levels of glycogen, but they have massive accumulation of lipids. And of course, CMA cannot degrade glycogen and it cannot degrade lipids. It only degrades proteins. So that let us trying to figure out what were the proteins that when they cannot be degraded by CMA, they will give us this phenotype. And through a bunch of studies that have been now published, we found that many of the glycolytic enzymes contain these CMA targeting motifs. And if they cannot be eliminated timely and they persist, for example, in the starving liver, what they are doing is continuously maintaining glycolysis, so you cannot even do storage of glycogen. And this is not something that the liver should be doing. And on the same token, you cannot repress lipogenesis. So these lipogenic enzymes that are also degraded among other pathways by this pathway keep producing lipids. And then even the perilipins that normally recover your lipid droplets and that they have to be removed, therefore lipolysis to occur, and they seem to be removed by CMA. So if you don't have CMA, you have also lower levels of lipolysis. So the phenotype that we see in these animals is a convergence of problems turning off both glucose and lipid metabolism at particular times in what that turning off seems to be necessary for the organism. But as I said, the nice thing of having different tissues and organs is that we also realize that in other organs, you also have contribution of CMA to modulate glucose and lipid metabolism, but in a very different way. So for example, a recent work from Susmita Kosik in the lab studying the process of adipogenesis has shown that in this case, what the CMA is doing is turning off glycolysis in order to accumulate lipids. So just to give you a glimpse of work, we have found, for example, if Susmita utilized an in vitro system for differentiation of pre-adipocytes into adipocytes. As you know, there is this very timely regulated process. The first thing that she showed us is that if she looks at changes in CMA during this process of adipogenesis, she can see a very nice upregulation of this pathway throughout this process. To see if this was important for adipogenesis, what she did is eliminating CMA and induce those pre-adipocytes that do not have CMA into adipogenesis and see what happened. So we compare with the control cells that by day 8, you already see these gorgeous lipid droplets that you can see here in blue. In the models, in the cells that you do not have the ability to upregulate CMA, you can see that there are barely any lipid droplets and there are little amount of lipids there. And similarly, if you look now at the tissues of these animals, of course, these animals develop adipose tissue, but it's a very deteriorated and abnormal tissue in which you can see a lot of proliferation and become very secretory, and the animals develop insulin resistance. So we were interested in trying to figure out, okay, what proteins need to be eliminated by CMA in order to go into adipogenesis. And through very different approaches, what Susmita demonstrates is that the first thing that you have to do is to modulate glycolysis. So as you know, when you have a pre-adipocyte and it's going to become an adipocyte, it needs an energetic burst. It needs to upregulate glycolysis, and then it has to shut it down, because if it doesn't shut it down, it will never accumulate lipids. So what happened in our cells that do not have CMA is that this is the upregulation of glycolysis in the controls at day 2, and then it goes down, and this is just seahorse. When you look at the cells that do not have CMA, this is what happened. They upregulate glycolysis, and look at the scales here, and basically they continue with this upregulation of glycolysis way above of the time that they should. So basically, because of the inability to remove these glycolytic enzymes at this very particular time, this pre-adipocytes continues maintaining very high levels of glycolysis, and consequently, they cannot accumulate the lipids. But then if that will be all, you will only need CMA at day 2. So why do you keep increasing CMA during the whole process? So she also found that CMA is required to stop proliferation. As you know, when you pass from a pre-adipocyte to an adipocyte, in addition of this energetic burst, the cells divide once, and then they have to stop dividing to start differentiation. So what she found is that this stop dividing occurs because MYC undergoes degradation by CMA. However, if you don't have CMA, as in this case of the LAMP2A cells, your levels of c-MYC remain very high, and these are 2 different models, and basically the cells keep dividing. And that's why in the adipose tissue, we keep seeing all these proliferative pre-adipocytes that cannot engage into differentiation. And then in addition, this program is very controlled, and then eventually you have to engage a whole transcriptional program that is gonna initiate and push into adipogenesis. When she compared the transcriptional program of the animals that have normal CMA or the ones that do not have CMA, so this is at resting conditions, there are already some transcriptional differences, but then if you compare what happened in the control versus what happened in the knockouts, there are many transcriptional changes that are not taking place in the animals with deficient CMA. So she utilized in-genitive pathway analysis to see what was the upstream regulator that could give us this lack of transcriptional changes. And basically, she identified that was persistent of signaling through the TGF-beta receptor. In normal conditions, in pre-adipocytes, TGF-beta receptor signal and phosphorylate SMAD3, and this is going to repress the progression into the transcriptional program for differentiation. However, eventually, when you need to progress CMA, remove this phosphorylated ESMA, take out this inhibition, and now you can progress into adipogenesis. In the cases that we don't have CMA, what occurs is that the levels of this ESMA3, as you can see here, persist elevated for longer time, and this kind of maintain a break that prevents the transcriptional program of adipogenesis. So as I said, this was just an example to illustrate that even reviewer 3 always want to find what is the single component that when cannot be degraded by CMA is going to give us the whole phenotype. What CMA is doing is proteome remodeling at different types of different parts of the proteome, depending on the cellular requirements. So here it goes from glycolytic enzymes to control of proliferation or to modulators of signaling. And this is basically what allows the whole progression of adipogenesis. And then, of course, sometimes I don't have time to tell you about all this, but sometimes it can be very exquisite. Actually, sometimes it's a very specific enzyme. For example, in the case of activation of hematopoietic stem cells, we have seen that the modulation of a very selective metabolic pathway by CMA is enough to facilitate engagement into activation. And then what happens when we don't have CMA in the whole organ? Because, I mean, so far I have given you organ by organ, but I also showed you at the beginning that we don't age only in one organ regarding CMA. CMA is going to get compromised eventually in all our organs. So work from Julio and Adrian in the lab have been very interested in using these CMA-incompetent mice and try to see how they respond to different metabolic challenges. So, for example, if you compare when these animals are young, wild type and CMA deficient, LAMP2 knockout animals, you maintaining them in CHO diet, early on there are not differences in weight. If you put them in a high-fat diet, in both cases they increase, so the response to high-fat diet is comparable in the control and in the knockouts that are in red. But then look what happened when you put them in a high-carbohydrate diet. So controls are not really gaining much more weight compared to the CHO. They can handle this carbohydrate pretty well, but if you don't have CMA, they really start gaining a lot of weight. We have been analyzing a little more this failure to respond to this high carbohydrate diet. And if you compare here the accumulation of fat, so in pink will be visceral fat, and in yellow is subcutaneous. And if you compare wild-type animals have an increase in visceral fat, but not really much adiposity. But look at the knockout. So they start having visceral and subcutaneous fat, and this is just the quantification. So to give you a summary of what happened with these animals when we challenge them, I told you that they become obese, they accumulate visceral fat. They start also having increased deposits of lipids in different organs. This is, for example, the liver, but we have also found it in skeletal muscle. So they start developing this lipidemia. When you look at the levels of circulating lipids, they are also increased in the knockouts. And then, of course, they develop, as you can see here, very dramatic insulin resistant and high levels of glucose. So basically, in a way, by challenging these animals with this high carbohydrate diet at very early ages, we can reproduce many components of the metabolic syndrome. So to summarize a little what else we have found in all these animals and organs, I show you that one of the mechanisms by which CMA decreases with age is this reduction in LAMP2A. If we mimic that reduction in a young animal, we see phenotypes of degeneration. I didn't show you, but they develop neurodegeneration, problems with retinal vision, also muscle dystrophy. They develop different metabolic phenotypes, as I showed you, fatty liver, metabolic syndrome, adipose dysfunction. These animals also have loss of stemness and immunosenescence. They have dysregulated circadian rhythms, and they also have higher propensity to malignant transformation. So, of course, when you look at these characteristics, many of them are characteristics that you observe in aging animals. Of course, I don't want to say by any means that loss of CMA is the cause of aging. We all know that aging is a result of many different drivers, but this is definitely this decrease of CMA with age is something that is not helping with the aging phenotype, and if anything, is accentuating it. So that kind of let us try to figure out, okay, how are we going to fix this? I think at least we are convinced that this loss of CMA that is going to occur in everybody, even under physiological conditions, is not something good. So can we fix it? So the lab has engaged in different types of approaches, genetic, dietary, and chemical. And I'm just going to give you a glimpse of what we have been doing genetically, because I think it's quite interesting. So in Mataset, junior faculty in the lab, decide that since we know that part of the problem is this decrease in the levels of LAMP2A once you reach mid-age, maybe she can create an animal in which these levels of LAMP2A can be maintained constant. Because the idea, we didn't want to create an animal that is a super mouse that has tons of CMA activity when they are young. We just wanted normal animals that then when the endogenous copy start going down, she can induce an extra copy of LAMP2A and then just maintain levels as when they were young. And then, of course, we got a little greedy, and because I have Y pass mid-age, we thought it's like, okay, what about if you start enhancing the CMA activity, or you activate this extra copy once the animals are already past midlife? Can we still recover activity, or at least maintain the activity that we have? So for sake of time, I'm just going to show you these two groups, the ones that have never seen a decrease in CMA in orange, and the control animals at different ages. Of course, when she did lifespan, I'm going to stop the video. When she did lifespan, so these are males and females. And as you can see here, there is a nice increase in maximal lifespan, but also in mean lifespan. And this is the one that I like, because that means these animals are healthier. So in collaboration with Rafa de Cabo, we've been measuring frailty and all these 32 parameters. But I thought that rather than showing you 32 different graphs, it was better to just show you the animals. So these are litter-mate animals that are 26 months old. And this is the wild type, and this is the animal that has never seen a decrease on CMA. Just by looking at them, you can see this guy is balding, has white hair. This one is shiny coat, how they behave. This guy is kind of hardly there, doesn't move much. This is the wild type. And look at this guy. He's all happy. He's moving around. I mean, I want to be that mouse when I become old, because in maintaining full activity. We have done numerous tests from behavior, metabolism, and all the parameters that we can see. This is much healthier, definitely, than this other animal that will never move. You can see it's wobbling, it's opening the legs, and it's compromised at all the levels. Just to give you related to metabolism, for example, we have done metabolic cages, and in orange is again the ones that have preserved CMA, and not only they are energetically better, but they also recover this circadian. If you remember, this has to be different in the day of the night. With age, you lose that, and now they are even recovering that circadian. We have looked at 1 million parameters. They have less fibrosis, less senescence. I'm more than happy to comment about that. But I just wanted to show you this glimpse as a proof of concept that if we found ways to preserve CMA activity as the same one that you have as when you were young until late in life, that might have a beneficial effect. So, of course, we are doing a lot of chemical development, and I'm happy to talk about that. But I thought just to finish this talk, I wanted to illustrate some of the efforts that we are doing with diet. And then just pick one of them, but we can talk about any others during the discussion. So one of the things that we have looked in collaboration also with Rafa de Cabo was calorie restriction. As you know, calorie restriction has been one of the paradigms that have been shown to more consistently improve health span in many different species. And even you have to produce a level of adjustment depending on the sex and depending on the strain. It has been shown to be beneficial in aging animals. So Marian in the lab, in collaboration with Susmita, decide to subject our animals to calorie restriction and then see whether that was enough to improve CMA activity. So basically, she got like wild-type animals, and these are the ones that we used to get from the NAA colony that have been 40% calorie restricted, maintaining balanced diet since they were 4 months of age. So if we look at the wild-type animals, and we can reproduce in vitro this decrease of CMA, we isolate lysosomes from liver, we provide radiolabeled substrates, and then we can see that compared to the lysosomes from young animals, as the animals become old, and these lysosomes are intact, there is not breakage, but they have lower ability to perform CMA. So now if we get the lysosomes from the 12 and the 22 animals that have been under calorie restriction, we saw this very encouraging increase in CMA activity. And we have 1 million more experiments that were done, but just to illustrate that early start of calorie restriction seems to be beneficial. And something that was very interesting is that you can transfer that beneficial effect. I mean, this was in animals, but if we get cells in culture, and we just culture them in the presence for 1 week, in the presence of serum coming from ad libitum maintained animals or animals maintaining calorie restriction. So we are just getting fibroblasts and we are just adding serum from these animals. As you can see here, remember, PANTA means activity. So compared to the are being maintained with the ad libitum serum. The cells that are maintained with the serum from the calorie-restricted animals have this beautiful activation of CMA. So then we wanted to know if we can also do this in vivo, but also if we can do this once you have this decrease in CMA. But instead of calorie restriction, we start thinking that maybe it's time to use calorie restriction mimetics, that, as you know, is this variety of compounds that modifying levels of acetyl-CoA have been proved to be beneficial and to make many of the effect of calorie restriction. So we use 4 different ones, but for sake of time, I'm showing you here one of them, the 646, that has been proved to extend lifespan. And we just wanted to know if in 18 months old mice, where we already see a decrease on CMA, just by injecting, for example, for 3 days, this calorie restriction mimetic, we were seeing any changes in CMA. I'm not going to show the quantification because I want you to see the images. So, for example, this is liver, pancreas, and kidney, and you can see the dots corresponds to CMA activation, and you can see very nicely how only 3 days after giving this treatment, you already have a very nice activation of CMA comparable to the one on the young animals in all the organs that we look. This is the kidney, this is the liver, this is the pancreas, and this is the kidney, suggesting that you can restore CMA activity with these caloric mimetic compounds. But then the next question is like, okay, these compounds and calorie restriction activate CMA. Is this part of the beneficial effect of these drugs or of these interventions? So in order to do that, Marianne, and because of the sensitivity to high carbohydrate diets that I have shown you before, decided to use the high carbohydrate challenge because it has been shown that when you have animals that are a high carbohydrate diet, if you gave these calorie restriction mimetics, they seem to be protected against the diet. So part of the beneficial effect of these mimetics is by preventing the damage that you have in liver and many other organs with this diet. So we decide to go ahead, and these are just maintained ad libitum. So this is the liver. We are staining for lipid droplets. And as you can see here, this is ad libitum. This is the high carbohydrate diet, or you can see this very nice accumulation of lipid droplets. But if you put the caloric mimetic, you see a reduction in lipid droplets. Sorry. So now, what happens if we do the same in animals that do not have the ability to upregulate CMA, in our knockout animals? So when you look here, not only our animals accumulate more lipids, but also the caloric mimetic lose their ability to protect. And we also have done it not only for liver pathology, but if you are not able to upregulate CMA, you cannot reduce the liver steatosis, fibrosis, the increase in mass, or even the changes in the adipose tissue. So this data kind of suggests that part of the beneficial effect of dietary interventions such as the calorie restriction, or in this case, the caloric mimetic, is at time by this upregulation or restoration of normal CMA activity in these all organisms. So just to summarize a little what I wanted to convey today, I hope I convinced you that this process of lysosomal degradation of CMA is important for quality control, but it goes beyond that. It has regulatory activities, and I have focused mostly on regulation of glucose and lipid metabolism in different organs. And I have shown you a little how this is different, for example, in the liver or during the process of adipogenesis. And I have also showed you how aging, but also different types of dietary challenges are inhibitory on the activity of this pathway and might contribute to accelerate some of these age-related conditions. And then I gave you a glimpse of some of our attempts to restore normal activity of this pathway, either genetically by restoring normal levels of LAMP2A in our transgenic animals that, if you remember, were healthier and in better conditions, or just different dietary interventions or dietary supplements, such as, for example, this calorie restriction mimetics. So with that, this is the most important slide. I want to thank all the members of my lab. As I say, I only know about lysosomes, so anything related to any organs or any physiological activity, it's really coming from their knowledge. And we have the luxury to work with very, very helpful collaborators that hold our hand through all of these projects. This is the people who gave us the money. And of course, I just want to give a shout out if you are interested in autophagy. We created 3 years ago an international network that is called Women in Autophagy, but is open to everybody. And the idea is to promote the careers of young investigators interested in autophagy. It's completely free, has a bunch of activities from workshops to meetings to conference to repositories. So you are interested, I encourage you to join this program. So thank you so much for your attention, and I will be happy to entertain any question.
Well, thank you so much for a very interesting presentation. And we do have some questions. First question comes from Elizabeth Whitcomb. She says, great talk. Did you look at the circadian regulation of CMA in the retina?
Yeah, so that's great. So we have we are doing all our retina collaboration with Patricia Boya, and we have been looking at retinal degeneration actually. But in the case of the animals, remember these are mice. So what we did is like we check at the changes in CMA during the different light and dark cycle. And it was interesting that, for example, rod and cones have different cycling time. They both cycle, but they cycle at different times. And then we couldn't find cycling in ganglionar neurons. So you probably know much more about this than me. I don't know what that means, but if it's of interest, I'm more than happy to continue chatting with you on the side. But we were kind of curious that we found those differences.
And Joe Mason asks, are there any common SNPs or other genetic variants of LAMP2A in human populations? And if so, what are the phenotypes?
Yeah, so this is very interesting. So mutations per se, there is only so far a disease that has been described that is called Dannon's disease. So this is it was described as a muscle vacuolopathy. And these are kids that when they are in their 20s, they die of heart failure. And the phenotype is mostly the presence of big vacuoles in the heart. And the skeletal muscle. And the interesting thing is that the mutations are in most of them are in the common region for this gene. So that will give you mutations in all. And this is a deletion. So basically you will not have LAMP2A, but you will not have LAMP2B and LAMP2C either. And what is interesting is that LAMP2A is important for CMA, but LAMP2B is important for macroautophagy, and LAMP2C important for RNA degradation in lysosomes. So it's like 3 autophagy pathways controlled by a single splicing. So most of the vacuolopathy can be reproduced in a family that has the mutation in the B. So everybody is assuming that that modification is in the B, but the patients will also have, the ones that have the mutation at the beginning will also have deficient CMA, and they will also have deficient in RNA handling. Regarding variants, and this is something that we've been doing in collaboration with Dolores Corella and with Jose Ordovaz in your center. So Dolores Corella, I don't do any genetics. They are the ones doing all that, but they have identified a variant of LAMP2A that seems to be protective against cardiovascular diseases, and other that it seems to be aggravating for what it's worth. So at the moment, we have to investigate at what level is modifying LAMP2A. And then there is also studies that we did in collaboration with UCISA that centenarian individuals, so progeny from centenarians that we have access in Einstein, then they have also a variation in LAMP2A, and that was specifically in the A exon. And this was only present in centenarians, but we haven't done much further to see what are the functional consequences.
Next question comes from Michael Lustgarten, and he sends along his greetings. Then he actually asks a two-part question. So he asks, when considering that the high-fat diet that you use contains the poorly fermentable fiber cellulose, if the high-fat diet was instead supplemented with fermentable soluble fiber, would the CMA reduction be reversed? And the second part of the question is, if so, would it suggest a role for gut bacteria on liver autophagy, or is that already been tested?
Hi, Michael. Yes. Great question. So we didn't replace with fiber, and we were kind of interested that the decrease that we see is much more pronounced with high carbohydrate diets than with the high fat diet. And in the carbohydrate diet, the fiber is better preserved. So, we have to do the experiment, but it might be in part also because of the fat. But you really hit it with the gut microbiota. So we have done, and these are ongoing studies. It's not that I don't want to share, it's that we're still analyzing them, but we have collected the microbiota from the animals with preserved CMA until late in life, the knockout animals, young animals, and all animals, and we are currently analyzing. So far, what they have told me is that there are major differences, but we haven't really met with the experts to figure out what they mean. But there are clearly differences in the microbiota that obviously can contribute to some of these metabolic changes that we see. I mean, it's not all about the circulating lipids or glucose, but we think that also even intestinal absorption that changes with age, we can prevent that. I mean, we can preserve it longer in these animals. So I think that might also contribute to the impact of the microbiota. But yeah, looking forward to see those results. We haven't finished that yet.
Next question is from Zhongdan Wang, and he said, a very interesting talk, and he thanks you for that. And then asked, you mentioned that retinoic acid receptor alpha inactivates CMA. I have two questions. One is the retinoic acid receptor alpha is expressed in all of the tissues, but with different levels. Do you think that CMA activation could be selective due to the different expression levels of RAR alpha in different organs and tissues? And the second question is, retinoic acid receptor alpha also is expressed differently in certain disease conditions, example, non-alcoholic fatty liver disease versus hepatocellular carcinoma cancer. Do you think the selective CMA activation could be either beneficial or detrimental, that is, inhibit high carbohydrate diet-induced fatty liver, but promote growth of hepatocellular carcinoma cells due to increased CMA?
Yes, so great questions. And I can tell you a little what we know about the retinoic acid regulation and why we think it's a bit more selective than just like the whole transcriptional program. As you know, retinoic acid receptor modulates more than 275 genes. And as you point out, the levels of expression are very different depending on the tissues. What we found, and it was not arbitrarily, but basically we find out that, as I said at the beginning, if you eliminate retinoic acid receptor, you have more CMA activity, but of course you have so many other changes in the cells that that's not doable. But in collaboration with Evrysdi Gabariotis, we have developed some small molecules that seems to be able to only modulate part of the transcriptional program of retinoic acid receptor. So now we have a paper that was just published recently that what we found is that these molecules, what they do is control the binding of, you seem to be an expert in retinoic acid receptor, so you know about the NCOR, that is a co-repressor of retinoic acid receptor, and that seems to be the physiological regulator of this inhibition on CMA. So we were lucky enough, or the knowledge of Evrysdi Gabariotis, that was the chemist, was so good that he was able to stabilize this repression only mediated by NCOR. And when we look at the transcriptional program, most of the genes that are under retinoic acid receptor are still changing properly, are still not repressed, but then you have like 32 genes, of which 12 of them we know there are CMA regulators. So it seems like this very discrete CMA transcriptional program, very similar to what happened with macroautophagy and TFAB. And then just by hitting that, we are able to have the beneficial effect of blocking that transcriptional repression without altering all these other important functions of retinoic acid receptor. Regarding the correlation with levels, we haven't found so far correlation between levels of retinoic acid receptor and CMA activity, but we have found a relation between the ratio of NCOR, the repressor of retinoic acid receptor, NCOR retinoic acid receptor ratio, completely correlates with CMA activity. And going a little further, and you were talking about disease, the way why we got interested in a disease that is called retinitis pigmentosa in collaboration with Patricia Boya is because we found that those patients have very low levels of CMA activity. And then there is beautiful transcriptome and proteome done, and they show that they have very low levels of NCOR. So basically, as I said, there is this correlation, and by using the drugs, we stabilize the little NCOR that we have. We get it to stay longer in the retinoic acid receptor, so then you have CMA activity increase, and in the animals, we were able to preserve the vision. So basically, we think that more than the retinoic acid receptor is the ratio of repressor and receptor, what is really correlate well, at least in most of the studies that we have done with CMA activity. And then for the second question, yeah, the disease conditions, we have looked a lot in the retina because that's where we find NCOR. We have also looked in Alzheimer. Actually, this is unpublished, but we also see a good correlation there. We haven't looked in the context of, and it's a pity because we have all those livers, so we should do that. So of fatty liver, because we've been inducing for different process fatty liver in these animals. So we definitely should look at the correlation there. But again, I think NCOR retinoic acid probably is going to be more informative. And then regarding whether too much CMA, I think one of the questions there is that if it can be detrimental. So far, we don't think, at least not through this signaling, we haven't found detrimental effects because remember, even the lysosomes are the ones doing the degradation and LAMP2A is required. The one identifying what has to be degraded is the chaperone. So the chaperone is the one making the call. So it doesn't matter that you put more receptor in the surface of the lysosomes. You don't have the chaperone identifying, okay, this is a protein that we want to degrade at this time. It's not going to degrade more than it should. It's very different to just trapping things. It's very selective. But of course, we see beneficial effect in disease or in conditions that you have less receptor because you have your poor chaperone there waiting for receptor to become available. So now, as soon as you put it, those proteins that have been tagged for degradation can be degraded. I think I covered most of the questions. Is that right?
Yeah, I think so. Okay. Alice Lichtenstein thanks you for a very clear presentation of an extraordinary amount of data and asks, for the high carbohydrate diets, is the primary carbohydrate source refined or unrefined, and have you compared different types of carbohydrate?
Yeah, this is a great question. So it's refined. And then what we have done is compare with fructose, a diet high in fructose, and it's still inhibitory on CMA. That one, the inhibition is less, but you still have all these consequences, and the animals are still very sensitive to high fructose diet. That's as much as we have done, yeah.
Jennifer Lee asks, or says, thank you for a beautiful talk. And for your calorie restriction studies, how did you decide on the 40% calorie restriction? Some people express concern that 40% is too high compared to the 20 to 30%.
Yeah, you are completely right. And I can tell you the truth, or I can make us look good. So I'm going to go for the truth. So the truth is that we started those experiments. You're not going to believe it. We started those experiments in 2005. And I was the one doing that project because I was still doing research in the lab. And then, of course, life happened. So I was not doing research. Nobody took the project until Marian took it. But in the meantime, because we collaborate a lot with Rafa De Cabo, you probably have seen his beautiful work showing that depending on the sex of the animals and depending on the strain, you also have to adjust. You always get beneficial effect, but you, as you point out, you really have to adjust the percent of calorie restriction that you were doing. In our case, the early studies are all with the NAA colony that was 40% across, and in that strain has been extensively proved to be beneficial. But you are completely right. If we change the strains, we'll have to change the percent of calorie restriction. And in a way, that's why we moved to the caloric mimetics, because we can control a little more the doses of and to see how much we need to regulate. And we are currently, as we speak, trying to do the same. That those studies were all in males, and now we are trying to do it in females because, as you know, sex also condition the percent of restriction that you need to get beneficial effect. But these are all very good points. We started the game too early, and nothing of that differences were known by then. Yeah.
Okay, and the last question of the day, unless another one pops up, but Chris Wiley says, wonderful talk. And says, you hinted at the reductions in senescence. Did you observe reductions in senescent cells or specific SASP factors among your aged animals?
Yeah, I love this question. So I have one of my graduate students is fully invested. Her whole thesis is about CMA and senescence. Because initially there was kind of a theoretical paper, I guess, because they couldn't do CMA. They theoretized, but they put this paper with the idea that CMA might be required for senescence. So we have done those studies. So if we block CMA, cells still become senescent. So it's not required for senescence. But then the interesting thing is that it might be required, and this is very preliminary, so she will kill me. She knows that I'm telling all about this, but we think it's required for the resolution of senescence. As you know, senescence is a physiological process for wound healing. It should go, cells get senescence and they get removed and life goes on. Unfortunately, when that removal doesn't occur timely, what happens is that these cells persist there. And as you point out, they start secreting these factors, these SAS, that is going to be negative. So far, what we have seen is that if you engage a cell that doesn't have CMA or very low levels of CMA, what happens is that they become senescent, but when we analyze, for example, their translatome, because as you know, senescent cells attenuate translation, or their SAS, because they have this secretory, they have a different secretome. Is still a SAS for most of the components, but it has some variations. And interestingly, it resembles much more the one that you see in all cells. So we think that part of the difference between the secretome of a young and an old senescent cell might be because of this difference in CMA. But the most shocking thing why we see less senescent cells in our animals seems to be related with immunoclearance and the ability to eliminate those senescent cells. Because if we block, for example, CMA just on macrophages, that's enough to have an increase in senescent cells. So we think that the role of CMA macrophages for senescent cell removal might be important. And as you get old, that gets compromised, and that might contribute, among many other things, obviously, to this increase in senescent cells.
Well, thank you so much. Thanks for staying on, first of all, to address all those questions. And thank you for a wonderful and very interesting presentation.
Thank you very much for all the questions. This was very exciting.